Good afternoon, everyone. We'll begin in about a minute or so. Good afternoon. So welcome everyone to our citywide psychiatry grand rounds for March 25th, 2022. My name is Raj Rasasingam and I am the CPD director for Department of Psychiatry. Thank you for joining us today. We're really appreciative of you being here to attend this event. Before we begin, I would like to do a land acknowledgement. I would like to begin with the acknowledgement of the land at which we work, study, and live. Before I read it, I want to take a moment to situate yourself and think about the land under and around you. We recognize that many Indigenous nations have long-standing relationships with territories upon which the sites of the Department of Psychiatry are located that precede the establishment of the University of Toronto. We acknowledge our presence on the traditional territory of many Indigenous nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, and the Haudenosaunee, and the Wendat people. Today, this land is home to many diverse First Nations, Inuit, and Métis people, and we're grateful to have an opportunity to live, work, and gather on these territories. We acknowledge that Toronto is covered by Treaty 13, signed with the Mississaugas of the Credit, and the Williams Treaty, signed with multiple Mississaugas and Chippewa bands. Since this event is a virtual gathering, a singular land acknowledgement does not capture the richness of our distribution across many locations. There are few places on earth that someone before us has not called home. So thank you. An evaluation, couple of housekeeping items. Evaluation link will be sent to the attendees after the presentation. Your feedback is always appreciated. Our appreciation to Dr. Jacob Bortsman and the Hospital for Sick Children for the joint citywide psychiatric grand rounds. That's the next grand rounds. It's going to be during our Future of Psychotherapy conference. So I'm going to stop sharing the screen, and I'm going to ask Dr. Vicky Sturgiopoulos, our Vice Chair of Clinical, to do the department welcome. Thank you.
Thank you, Raz, and thank you everybody for joining this month's Citywide Grand Rounds. As you recall, this is one of the initiatives of our new strategic plan focused on integration to foster greater collaboration between our sites and greater learning from each other and learning together. It is my great pleasure to introduce Dr. Sunita Monga, the Interim Chief of Psychiatry at Sick Kids, who's hosting Grand Rounds this month. Sunita.
So thanks, Vicky, and warm welcome to everyone on behalf of the Hospital for Sick Children. I'm really happy to be introducing Dr. Jacob Bortsman this morning. Dr. Vorstman is actually originally from Netherlands, and he completed his MD, his PhD, and his child and adolescent psychiatric training in the Netherlands. And he started off his career in the Netherlands, and he joined the Department of Psychiatry at the Hospital for Sick Children in 2017. And he's been a very active clinician and researcher within our department since that time. Dr. Jacob Bortsman has a special interest in working with children with rare genetic disorders. In fact, Jacob developed the developmental assessment of genetically susceptible youth, or what we call the DAGSI clinic here at the Hospital for Sick Children. This is the first child psychiatric clinic whose objective is to assess children primarily because of genetically inferred risk rather than behavioral concerns. And within the DAGSI clinic, Jacob and his multidisciplinary team integrate research and clinical care with a focus to shed light on the prevalence of neurodevelopmental outcomes in children with genetic risk variants and to develop and validate measures of prodromal features for this specific high-risk population. In addition to this kind of work, he has also developed a smartphone application for the measurement of social and communicative behaviors, which is currently being implemented in several studies, thereby generating the first data of this kind. Jacob has an international collaborative analysis of the largest data set of 22Q subjects worldwide. And he has a very impressive and extensive publication list, including in publications in many high impact journals such as Nature Medicine, Nature Reviews, Genetics, etc. And he also has published a number of book chapters and co-edited one textbook. In addition, Jacob is actively involved in a number of levels of teaching, and he's an inspiring mentor to child and adolescent clinician scientists. And finally, Jacob was appointed as the inaugural Psychiatry Associates Chair in Developmental Psychopathology in 2020. So it's my pleasure to welcome him here, and he will be speaking to us about genetics and precision mental health, the challenges and opportunities.
Thank you, Sunita. This is really wonderful, and I'm a bit at a loss of words. Thank you for your very nice introduction. And I want to say, I hope the screen is what it should be.
It's coming up, yeah.
Yeah, I'm just not seeing my, wait 1 second. There we go. Is it good?
It's good.
Yes.
All right. Thank you so much. All right. So thank you for the introduction, and I want to say that it's truly an honor and a pleasure to give this presentation. And I also want to say that what a blessing and what a fortune it is to be able to work in this environment with all the colleagues at SickKids, but certainly also across U of T. So thank you for attending this talk. Let's see if I have some disclosures to make. And the other disclosure is that I have a few pictures of this, one of my favorite cartoon characters. There's no actual reason for that other than I just like it, but you can maybe, because all of you are psychiatrists, maybe you find reasons, but I don't know of them. Anyway, the objectives here is to talk with you about genetic findings in our field, how they already influence our practice, what the challenges are, and what the opportunities. And so let's first have a look on what we already know. And I want to start with a short introduction. I know not everyone is as familiar with genetics. So a distinction very roughly we can make if we think about genetic variants is between common and rare variants. So if this is the entire population, you can take a variant and you can count the number of individuals with that variant. And let's say you find 30% of the individuals have that particular variant. And so intuitively, you can understand that if so many people have that variant, if it's so common, then it's unlikely that that variant has a large effect on disease vulnerability. You can do it again for another variant and another. And so schematically, just to show that there may be a group of individuals in the population that have many of these risk variants by chance. And so the thinking about common variants is that their effect on disease risk is cumulative. Individually small effects, but cumulatively the effect can become relevant. And in contrast, you have rare variants, and because they are so rare, their impact on disease vulnerability can be relatively large. And so by and large, there's no reason to think that these models are mutually exclusive. They're not. And also the distinction between common and rare is somewhat artificial. In reality, there's a spectrum from very rare to common. But it's helpful as a distinction to kind of hypothesize their effects. So if you look, for instance, in autism spectrum disorder, ASD, and you take a population of individuals with this condition, we know that there is a small proportion that where the ASD is, you can explain it by the genetic variant of a known disorder. So for instance, Fragile X or tuberous sclerosis complex. And then there's another fraction of about 5 to 10% where we know that are copy number variants. And here the work of Steve Shearer and his lab has been of paramount importance in the past couple of, let's say, 10 years. Decade, the role of single nucleotide variants or sequence level variants has been clarified in a proportion of individuals. And then there is also some individuals where there are there are recessive events that occur. But overall, the picture that is emerging is that in about 20 to 30% of individuals with autism, if you apply the right methods of genetic testing, you can actually identify a cause or a contribution to the phenotype. And so, you know, this is evolution. I like to call it a revolution, because when I started my residencies in psychiatry, this was just at the start of the millennium. If these are individuals with autism, we really did not know what was the cause of the autism. And of course, you're all aware of all the harmful and theories that were still even to the present day. You can still hear them, those theories. And so in between 2000 and 2010, the discovery of deletions and duplications that can cause autism, like I said, in Steve Shearer's lab, amongst others, and then more recently, the single gene variants that are found. And so the picture that is emerging is that autism is very genetically heterogeneous. So the same phenotype can have many, many different genetic etiologies. And the same is true for intellectual disability, or ID. So there's known genetic disorders, CNVs, and a contribution of single gene variants as well. And this is just kind of a very high level overview, but to show that today in people with intellectual disability, again, applying the right methods, we can find a cause in about 50 to 60% of these individuals. For schizophrenia, the story is somewhat different. The emphasis of the studies have been on common variants. And so what you can do at any location in the genome, you can see if there is variation of that particular base pair, if you wish, of DNA in the population. And we call these locations, we call them single nucleotide polymorphisms, or SNPs. And so again, you can count in a population the number of individuals that have a particular SNP, and you can compare healthy controls to patients, in this case schizophrenia. And if this difference is significant, you can test that statistically, and you can come up with a p-value. For instance, this SNP may have a p-value of 0.01, and maybe you're enthusiastic about it, but then you forget that we're actually looking at these SNPs across the genome. So we're not testing 3 SNPs, we're testing 500,000 or sometimes even more than 1 million SNPs. And this is why we call them genome-wide association studies, or GWASs. And obviously, if you test 1 million SNPs, you have to correct for multiple testing quite drastically. So the threshold certainly is not a p-value of 0.01, but it's generally accepted to be somewhere below p times 10 to the minus 7th or 8th. And so here are findings of the first iteration of a GWAS for schizophrenia. And so we're interested in seeing those SNPs that are exceeding that threshold. And so these are significantly associated with schizophrenia. There were only a few of them in this first iteration of about 7,000 individuals. And then the second iteration was in about 12,000, about 11 to 12,000 individuals. You can see already that generated more significant results. And then the last iteration was in 35,000 individuals with schizophrenia and over 100,000 controls. And that generated more than 100 robustly significant hits. But in schizophrenia, there's also studies done on the role of copy number variants, and this is work by Christian Marshall, again from Steve Shearer's lab, showing the involvement of a whole list of genetic variants, copy number variants, I should say, with schizophrenia. And so the overall high-level summary is that for schizophrenia and autism, for autism, there are more studies reporting on an increasing list of rare variants. So that's depicted here compared to the role of rare variants in schizophrenia. Whereas in schizophrenia, there are more findings that show the role of common variation that cumulatively taken together explain about 7% of the variants. Now, whether this difference actually reflects a different genomic architecture, if you wish, between autism and schizophrenia, that remains to be seen. It could also be an artifact due to the fact that there are simply more GWASs done in schizophrenia, and there are more sequencing studies that have been done in autism. So time will tell. Now, in the field of genetics, we have been quite obsessed, I will say. I use the word obsessed because the term came back all the time, to elucidate what has come to be called the missing heritability. And essentially, this is the difference between the predicted role of heritable factors predicted by twin and family studies and the actual effect size that sort of the explained variance that is explained by actually identified genetic variants. And so there is a discrepancy between both metrics. So the past 2 or 3 years, another discrepancy has come to light, and this has been aptly called the missing diversity in human genetic studies. And I wanted to talk a little bit about it because I think it's important. You can see the scope of this issue in these figures from this Cell paper. And what you see is that in GWAS studies conducted so far, there is a significant over-representation of individuals with European ancestry as compared to GWASs on people that are of non-European ancestry. Now, you may ask, why is this even relevant? Because after all, the world population is one big family, and we all have the same genes. Well, the reason is not so much the genes, but the ancestral differences in genomic architecture that may exist. And this is especially for GWAS relevant. And so one thing to realize is that the SNPs, the single nucleotide polymorphisms that are driving the association signal of GWAS, they may not be the actual causative variant. They may just be a variant that is close to that causative variant, and we call that a tag SNP. And so the issue is that causative variants in our genome may not be tagged by the same SNP in European people versus African people versus Asian people. And so, and even more so, there may actually be also difference in the actual causative variants. So in addition, it could be that in European people versus Asian versus African people, there may be different causative variants. And so obviously they're not being detected in a GWAS that only or predominantly looks at people from European descent. Okay, so this is particularly relevant for the polygenic risk score, and I'm sure many of you have heard about this polygenic risk score, and I'll attempt to explain it to you briefly. What I'm showing here is another Manhattan plot showing the GWAS results, and this time it's autism spectrum disorder. It's a study in 2019. And the polygenic risk score doesn't only look at the SNPs that are genome-wide significant, so the SNPs above this line. It actually looks at SNPs that are also subthreshold, like, sort of nominally significant before testing, before correcting for multiple testing. And the thinking is that in this group of SNPs, there are many, many SNPs that are actually true positives, even though there's also a proportion, we know, that are false positives. But taken together as a group of SNPs, there is a true association signal if we just take them and consider them together. And so the way to do that is to apply to each of these single SNPs a weight and the weight is proportional to the strength of the association in this GWAS. So the stronger this SNP is associated, the larger or the heavier the weight. And what is the polygenic risk score? Well, it's simply the sum of these effect sizes, the sum of these weights for this set of risk alleles. So the thing to remember is that you can simply calculate a score based on this set of risk alleles for each individual in a population, as long as that individual wasn't part of this GWAS, right? And what you get is a distribution of polygenic risk scores. So every dot here is an individual with a certain score. Some have very high score, some have very low score, but the distribution actually is a normal distribution. Now, if you do that again, if you take a second set of individuals, and these are individuals with autism, what you'll find is again a normal distribution, but it's higher. And so there is a difference between the two distribution. And that difference is the effect size of the polygenic, is sort of the explanatory power of the polygenic risk. Okay, so if we bring these elements together, the polygenic risk score is derived from GWAS, right? Case control studies that I just explained. And I also showed to you that at this point, the vast majority of GWAS studies has been done in populations that are predominantly of European descent. And so if we apply the polygenic risk score derived from the GWAS in a new cohort of people that are from European descent, we actually have a pretty decent explanatory power of the polygenic risk score. But it shouldn't come as a surprise that if you apply the same polygenic risk score in a, let's say, a population of people, in a cohort of people from African descent, that effect size is substantially lower. How much lower? Well, substantially. This is a study by Duncan et al., but there are other studies that show that the difference here, for instance, if you apply the same PRS polygenic risk score in a African ancestry, it's less than 50% of the original explanatory power. So what's the solution? Well, at least part of the solution is just running more GWASs, but this time being more inclusive and mindful of a representation of ancestry of different populations across the world. Unfortunately, that's being done as we speak. It just will take time before we have those data. So this is clearly a challenge that we have at present. But as is often the case, there are also opportunities. And I want to highlight this one. This is a study that looked at rare variants in populations in Uganda. And this is a study involving 6,500 individuals. And what I really find striking, and this is just one example, there are more studies if you look for them, is that this study identified many rare genetic variants that were unique for this population. So it means these variants would not have been found if we would continue to look only, let's say, in cohorts of people from EU descent. So in other words, we just learn more if we diversify our studies as we should. So I want to shift gear a little bit and talk about clinical translation. So essentially the question here is, how nice and interesting is it that we make progress in identifying genetic etiologies, but how is it relevant to our clinical practice? In other words, to what extent do these findings contribute to better care and outcomes of patients? Because that's in the end what we are doing it for, right? So at the very minimum, this knowledge of genetic etiology provides diagnostic clarity, and it's useful information for genetic counseling. And I think you're all aware of it, but it's often trivialized as not important. And I want to argue against it. Telling parents that there is a genetic cause for their child's, let's say, autism or intellectual disability or schizophrenia may be extremely important, even if there are no other implications for treatment. And the same accounts for genetic counseling. And so in this paper, we worked out some examples of recurrence risk. So what if there's a child with autism or a family member, in this case, a mother with autism in the family, what is the probability that the child or the brother or the next person in the family will also have autism? And the point we're making here is that if you don't have genetic findings, there's there's actually not much you can say other than that the risk is increased, but you don't know to what degree. Now, if you have genetic findings in all of these examples, I'm not going to discuss each one of them independently, but in all of these examples, the genetic findings will provide you a lot more information and you can be a lot more helpful. So I'm thinking of a child that I was following back in the Netherlands with autism, and this young man had a older sister, and at some point the family came to me because that sister was about to get married and was just worried with her future husband about the probability of having a child with autism. And so we recommended genetic testing and actually found a pathogenic variant in a gene that explained the risk, the fact that the brother had autism, and that variant was de novo, meaning it only existed in this brother. And so this information was hugely reassuring for the sister and her soon-to-be husband. Okay, so in some instances, knowing the genetic etiology can also guide treatment choices. And that's emerging. It's not often, but it's certainly increasing. So this is a study that looked at this particular question, just under 100 individuals with ID that had positive genetic findings. And this study found that in 7 of them, the findings were actually helpful information for the treatment. And in 40 of these individuals, there were referrals that were medically significant and relevant for follow-up, let's say, for instance, to cardiology or to nephrology or to urology and so on, based on these genetic findings. And then in many families, just knowing the diagnosis was helpful to allow them to connect to other families or to networks or support networks and social media and so on. So that's for sort of the low-hanging fruit in a sense, but on a higher level, knowing the genes that are involved in a psychiatric condition provides insight or can provide insight in the biological pathway. And these biological pathways may be appealing pharmacological targets. And I want to talk about that a little bit as well. But before I do so, I want to highlight another challenge. And the challenge I want to talk about is pleiotropy. And so what do I mean with this? Well, I show you another overview of deletions and duplications that are associated with autism. And just to highlight the fact that the penetrance of these deletions and duplications is virtually never 100%. What it means is that if you take a group of individuals with, let's say, a deletion of 1q21.1, you'll find an increased prevalence of autism, but that increased prevalence is not 100%. In this case, it's 8%. So that's the first thing. This is variable penetrance. That's the term we use for this. The other thing to note is that it's virtually never only autism. There's also intellectual disability, there's ADHD. There really is not a good reason to put ASD first or ID first in these examples. Could also start with ADHD. It's really random. And then there are other brain-related phenotypes as well as phenotypes that are not related to the brain. Let's say, for instance, heart defects or kidney abnormalities. Now, you may say, rightly so, well, that's not unexpected because these are copy number variants, and I know my understanding is that these copy number variants often affect more than one gene. So, if you have several genes, you shouldn't be surprised that there are also several phenotypes. But the fact of the matter is that we are seeing the same pleiotropy and variable penetrance. We see them also in those single gene disorders that are associated with autism. And then still, I suspect that some of you may say, academically interesting and so on and entertaining in a way, but really this is not a clinically significant problem. It only happens in a rather small group of individuals with these exceedingly rare disorders, right? And I'm going to challenge that view because the context, the broader perspective is that it's increasingly clear that mental and physical health is often comorbid, and the comorbidity is substantial. And this is a study in, I believe, 34 million Americans. And this is another study, more recent, in 2 million New Zealand population citizens. They were followed up for 3 decades and showing the same thing. Having a mental disorder increases the risk of physical comorbidities. And so these findings, they beg an important question, and that's how do we explain that comorbidity? And so obviously it's likely that at least part of this may be explained by sort of secondary relations, like stress from having a physical condition may increase the likelihood of developing a mental health condition. Having a mental health condition may increase the likelihood of, for instance, decreased self-care in some individuals. That could then increase the risk of physical health conditions. But I want to submit here that there's really good reasons to believe that at least part of this observed comorbidity may stem from pleiotropy. And what I mean by this is that it stems from shared genetic causes. Let me explain a little bit more. So here are some examples showing that we are increasingly successful in identifying genetic variants, causes and contributions to these conditions, and just a few examples. But we should not assume that these variants that we are identifying are specific to these conditions. In other words, that these variants, a third of the people with congenital heart defects, we identify genetic causes. We shouldn't think that these genetic causes are always only causing congenital heart defects. That's not very likely. And indeed, to expand a bit on the example of CHD, congenital heart defects, this is an exome study showing that in patients with congenital heart defects, there is an increased enrichment of patients with rare genomic variants that are associated with these heart defects. But that significant difference that was reported in this study actually remained significant if they looked only at genetic variants that were hitting genes that are important for neurodevelopmental disorder, NDD. All right, and indeed, again from this paper, looking at those genes that are involved in these variants, they saw, not surprisingly, that these genes are likely to be highly expressed in the heart, and you can see that here. But what is more interesting, I think, is that the same genes are also more likely to be highly expressed in the brain. You can see that on the y-axis. So it's this upper quadrant here that they highlight, and for good reason. So with Ni Wang, and she's the genetic counselor on our team at the Autism Research Unit, we examined the genes that are currently on sequencing panels for congenital heart conditions and the cleft palate and craniofacial. And we found that 10% of the genes that we are looking for when we want to know the explanation for the congenital heart defect, 10% are also involved with neurodevelopmental outcomes, and 20% of the genes that are on the panels when we look for cleft palate and associated craniofacial abnormalities. And together with Daniel Baribeau, our clinical research fellow, we looked, we want to know the scope of this issue in real data from SickKids. And so we looked at the genetic tests that have been ordered, both chromosomal microarrays as next generation sequencing, a couple of years, you can see it here. But all these tests, we only took those tests that were ordered for physical or congenital indications. So, congenital heart defects and cleft palate and those type of things. And then we looked to what extent the positive results were also associated with neurodevelopmental disorders. So I'm just merging those two types of tests here just to get to the results. We found that, or she found, I should say, that 5 to 10% of all the genetic tests that are ordered end up revealing also an association with a neurodevelopmental disorder. And that actually amounts to about half of all the positive results. So that's actually pretty significant finding, I think, pretty relevant, because why is this relevant in our clinical practice? Well, because in SickKids and elsewhere, genetic testing is increasingly becoming a routine part of the diagnostic workup in these kids born with these congenital abnormalities. And we are, as I've shown, more and more, successful in finding a causal genetic variant. And so we can sit down with parents and say, we understand, we can explain why your child is born with a cleft palate, for example. But more and more, we have to extend that conversation and say, but the gene that we found or the genetic variant that we identified is actually also associated with an increased risk to develop a neurodevelopmental or psychiatric disorder. And the truth is that currently we're not doing anything, at least not systematically, for these families. We just kind of share this information and we send them home. And so there is clearly a challenge here, but again, also an opportunity. And I've shown you that this actually happens, having to extend that conversation in 50% of these results. I want to argue that the opportunity here has to do with our ambition towards precision medicine. And I'm going to take the example of the 22q11.2 deletion. And as you know, this particular deletion is associated with schizophrenia and other neurodevelopmental phenotypes. Dr. Ann Bassett here at CAMH has done a tremendous amount of work internationally in this area. And so a typical trajectory nowadays in 2022, I'm not talking in 2000, right? 2022, typical trajectory is this one. A child is born and a congenital heart condition is discovered. And this immediately prompts genetic testing. And so in this kid, we'll reveal a 22q deletion, typically in the first months of life. And so at that particular age, the child is 3 months of age, we can actually tell the parents that the child is at risk for neurodevelopmental outcome, autism, ADHD, intellectual disability, anxiety. And actually we know that 1 in 4, 25%, 1 in 4 of these individuals will go on to develop schizophrenia. And so I want to argue two things. The first one is that the identification of a genetic variant implies that this child is genetically at risk or vulnerable, if you wish, for these outcomes. That is clinically actionable information. We cannot just leave that there and not use it. The other thing I will talk about is that genetic etiology can also inform treatment options. So this is, and Dr. Sunita Monga already alluded to it, this is why we started the DAGSI clinic. This is Dr. Jane Summers. She's our psychologist and the coordinator of the clinic. DAGSI stands for Developmental Assessment in Genetically Susceptible Youth. And so we target kids that have an identified genetic risk for neurodevelopmental outcomes, broadly considered. So autism, intellectual disability, psychosis, ADHD, and so on. We provide a comprehensive multidisciplinary assessment of these children, and our recommendations and assessment take into account what we know, what is known, or what can be known about the impact of that specific variant in that particular child. And so we use the DAGSI clinic as a platform for various research projects. We want to identify early biomarkers for neurodevelopmental trajectories and outcomes. We study modifying factors, so what in these kids can modify the risk of developing autism. And we're kind of working on a project to examine early interventions, because early interventions is another opportunity here. This is a recent Lancet paper co-authored by Evdokia and Agnostu and Peters and Mary, amongst others, and it outlines the future of care and research in autism spectrum disorder. And I want to highlight this particular figure because it sort of visualizes factors that modify outcomes. But the starting point, and I'm highlighting this myself here, is the genetic vulnerability. The question is, when do we see these kids? You ask yourself, when do we see these kids? Here? No. We're seeing these kids typically either because there are developmental concerns, but quite frankly, more often we see these kids because behavioral aggravations have developed because in the interaction with the environment. So think about social exclusion or bullying in school or academic failure and so on. And so that's also the time when we, for the first time, think about intervention to help with these situations. So the opportunity we have is that at least in a subset of children, we can by virtue of knowing their genetic risk, we can see them before these problems emerge. And so we can start thinking about something that we have never thought about in psychiatry, which is prevention, or at least mitigating strategies that can be applied early in life. The other opportunity refers, and I mentioned it before, to the biological information conveyed by the genes. And so here's another list of genes that are associated with autism. And I'm using a beautiful picture again by Daniel Baribeau from a recent publication, just to show that these different genes, they converge into biological pathways. In this particular example, and these are relevant for autism, it's the synaptic function, it's the postsynaptic scaffold pathway, and then chromatin remodeling and regulation. Why is this important? Well, each time we identify one of these, many etiologies of autism, even though we may identify a cause that is only relevant for a tiny fraction of the entire population of children with autism, or people with schizophrenia, or people with intellectual disability, we are actually chipping away from what we don't know about biology. In other words, we learn about the biological pathways, because if we put together these genes, as I've shown in the previous slides, we are actually elucidating meaningful and relevant pathways. And these pathways, they are the starting point or the target for the development of pharmacological compounds. Is this theoretical? No, not at all. This is an overview of clinical trials in autism. You can see, for one, that the number of trials have increased in this period of time, but also to show that the number of trials that are looking at novel compounds instead of repurposing is, proportionally increasing. And many of these compounds are informed, the selection of these compounds is informed by genetic findings. And, I didn't have time to update. It's time to update this table for sure. But again, I borrowed from Daniel Baribeau's work and just showing you a nice overview of currently ongoing trials, a lot of them, where the choice of the compound, the molecular target, is at least partly and sometimes completely informed by genetic findings, the biological knowledge that we derive from our genetic studies. Another point I want to highlight here is that there are certain studies where the choice, the selection of the population is informed by genetics, and that speaks to the principle of genetic stratification. So that's another way of using genetic data. The idea is that you use genetic markers that have been found and that you can use to distinguish distinguish the likelihood of treatment response or maybe adverse effects to inform your treatment choices for particular patients. So again, this is not utopia. This, for example, is hot off the press. It's exciting work by our colleagues at CAMH and across various U of T, other U of T institutes. And just showing you one illustrative result. There's a lot more in this paper, but they examined in this paper to what extent using genetic information can inform genetic information that informs on the likelihood of side effects and treatment response, to what extent using that information can improve outcomes in the pharmacological treatment of depression. And you can see that there's this difference. It's not significant. That's a power issue. But then there was another study done, a very similar design, a larger study in the United States. And you can see the accordance between the study findings. So a shout out to Daniel Muller here in the audience. I'm sure if you're interested to know more about pharmacogenetics, I would strongly recommend that you attend his grand rounds on April 22. He'll talk about that. Another example of something that is happening right now, just to kind of illustrate that this is not just theoretical academic entertainment. This again is the 22q deletion, and we now know that many of the genes that are involved in this deletion are important for glutamatergic signaling. And so the thinking is that a disruption of glutamatergic signaling is important and maybe help to explain the observed psychopathology. So the neurodevelopmental disorders and schizophrenia. And so it's only logical to think if there is a compounds that can regulate glutamatergic signaling, and there are several, but there's one here. Could it have a positive effect on these psychopathological signs and symptoms? And so that's the study we are, it has been started already. Actually, it's a multi-site study, international phase 2, and actually SickKids is in the lead of this exciting trial. I want to highlight one other challenge, and we're coming to the end of this talk slowly, but it's this challenge of variable penetrance. And so looking at these rare variants and the copy number variants in particular, what you can see is that most of these rare variants are not associated with one specific neurodevelopmental phenotype. It's often a range of different phenotypes. However, that doesn't mean that these variants or the impact of these variants is non-specific, because actually the data is showing that that is not true. If you look at a different variant, this is the duplication of the same region, you can see that the profile of impact is not the same. And this is true across a range of other CNVs. And they all seem to have a somewhat specific or characteristic profile of impact. A clear example, not in this chart, is not all of these CNVs have this impact on the risk of schizophrenia that we see in 22q deletion. For example, the duplication doesn't have that impact, right? So we can have a closer look at the issue of variable penetrance when we consider the phenotypic impact in a dimensional way, and IQ is a really nice candidate to do that. So in the general population, IQ is normally distributed. The mean is 100. And what we see in 22q is a similar distribution, but the mean is shifted to the left with about 30 points. So we think that that 30-point left shift is the impact of the deletion. But we still see a distribution. And so it means, if you see a couple of 22q patients, you may see some of them have an IQ in the sort of severe or moderate ID range, whereas others will have IQs in the normal IQ range, right? And how do we understand that? Well, in the general population, we obviously see the same sort of, variability in IQ, and we know now that that's for a big part under genetic influence of many, many common variants. And so the thinking is that in the 22q deletion population, it's the same. It's the rest of the genome that impacts and modifies the outcome in the presence of the 22q deletion. So we've looked at that. This is Anya Fiksinski. She now has a PhD. She has worked together with Ann Bassett and me and others. And this is actually Ann Bassett's sample. She tested IQ with the same measure in parents and in offspring, their offspring with 22q. And so I leveled the two distributions, so basically taking into account that 30-point left shift, right? I already accounted for the impact of the deletion. And then here are the findings in 22q. And in orange are those with intellectual disability, according to the 70 cutoff as a cutoff. And so I gave parents the same color. So the parents have the same color as their offspring, just to show, sort of visualize that if you take, if you account for that impact of 30 points, you see that there is an intact correlation between parental IQ and children IQ, just as we see in the general population. Now, this doesn't provide evidence, of course, that it's genetic. It could be environmental. So we need our genetic studies, and we have genetic studies to show that. And again, I already explained, you can use the polygenic risk score using GWAS. You can sort of, you can summarize the impact of many common variants cumulatively as in the polygenic risk score, and you can calculate that in any individual. So what we did, and this is Robbie Davis, who's a postdoc and moved on to Oxford. He was working in Steve Shearer's lab. And we found that we did that in 22q people. And so what we found is that there was a intact correlation between the polygenic score for IQ derived from a GWAS and the IQ in people with 22q deletion. And it explained about 3.7% of the variance. And that's a small explained effect size, but it's very similar to what we found in the rest of the population. And we did the same for schizophrenia. And this was, by the way, also done by Ann Bassett. And we saw that there was a difference in the distribution of polygenic risk score in 22q people with and 22q people without schizophrenia. These are just my last slides here. You can look at the distribution of the polygenic risk in any given group of people, and you can reason that those that are in the extreme end, at the left, they have the lowest risk because they have the lowest polygenic risk score, and you can compare them to those that are at the other tail end and they have a higher risk. And indeed, these are actual findings from autism. If you plot, you sort of look at the risk associated with having a polygenic risk score on this end, and then you walk towards the other end, you can see that the associated odds ratios increase in a very smooth way. And so if you think about clinical application of the polygenic risk score in 22q, you have to look at positive predictive value. And the positive predictive value is the number of individuals that are actually true positive amongst those that have a positive test finding. There's another formula for this, and I'm not going through it, just to highlight that there is this notion of prevalence. And what it basically says is that your performance of the positive predictive value of a test is also dependent on the baseline prevalence of the population in which you use the test. And this is really important because we took the polygenic risk score, and here, just to contrast, we applied that polygenic risk score to generate positive predictive values to predict intellectual disability. And we applied it in the general population, and that's what this is showing. This dotted line here is the baseline prevalence of ID, intellectual disability, in the general population. It's about 2%. And so what you see if you walk in the distribution from left to right, you see that there's not much happening. This positive predictive value is not distinguishing from baseline risk. Now, in 22q, the baseline risk for intellectual disability is 42% in our sample. So that baseline risk is considerably higher. So what happens if you calculate your positive predictive values based on that baseline risk? Well, then the polygenic risk actually becomes really informative, and you can distinguish between a group of people with 22q deletion where you can predict that their risk of having intellectual disability is, let's say, 62%, from a group where, you know, the lowest 10%, where the group where the predicted risk is, let's say, 24% or so. The same is true for schizophrenia. And so, arguably, and this really brings me to the last slide here. Arguably, these findings are still not sufficient to alter clinical practice, but I'm going to say that we're getting closer, and it's becoming really closer to clinical relevance. So, in conclusion, what I've tried to discuss with you today, there is just gigantic advances in our understanding of the genetic contribution to several psychiatric and neurodevelopmental conditions. There are challenges. I mentioned ancestral diversity, pleiotropy, variable penetrance, but I also highlighted that these challenges are also opportunities, particularly for precision medicine. And I guess my take-home message mostly is that these findings already influence clinical practice, if you want it or not, and it will inevitably impact more if we go forward in time. So with that, I want to just thank all the people I work with. I'm not going to name all of them. I've named a few of them. And the funding, I'm very fortunate. And I already mentioned at the start that it's just such a pleasure to work at the University of Toronto. Thank you.
Thank you, Dr. Vorstman. This was a wonderful presentation and very thought-provoking, and really it makes us think a little bit more about the research aspects, as well as clinical translation, as well as advocacy. So there's many different aspects that I really love the way you weaved everything into your presentation. So we have some questions that have come up, and you know, with autism, there's always different views and points. So there was a question that someone was asking about in terms of there've been many attempts to look at specific genetic findings that are responsible for particular autistic traits, eye gazing, you know, etc. You know, have there been attempts to look at specific traits instead of looking at autism as general? Has there been an attempt to look at that?
Yes, definitely. There's quite a bit of literature on it. Let me see if I can summarize this on a high level. The highest level is probably that there's emerging and converging findings that these genetic variants may come with specific profiles. And so some of these variants, even though overall the picture is that of autism, so they meet criteria for autism, there are certain genetic variants where, for instance, the repetitive behaviors are more prominent, and there are other genetic variants where maybe the issues are more, let's say, in eye contact and so on. So there's a lot going on. The jury's still out, but yeah, this is where it seems to be showing that there is a sort of gene-specific effects. Yeah.
Yeah. I mean, just a follow-up of that they were saying was, they're a clinician that sees complex medical and cognitive problems, child diagnosed with developmental delay, et cetera, and other epilepsy, et cetera. And they find it difficult to understand how autism could be applied to some of these different traits. I mean, any thoughts on that? I mean, and that's a complex discussion in
Sure.
itself.
Yeah, sure. So I'm not going to deny it. If children are complex, well, children are complex for sure. My own children are complex, if I think about it. But anyway, children are complex to start with. But certainly, children with complex medical disorders makes it even more, complex. And so the comorbidity between intellectual disability and autism. If you have a child that is really low functioning intellectually, it becomes really difficult to make the assessment for autism, but it's certainly not impossible, right? So what you have to do, generally speaking, is you have to think about what is sort of the age equivalent that the child is functioning on. So let's say you see maybe a 10-year-old, but the intellectual disability is such, or the cognitive level is such that the child may be functioning on a 3-year-old level, right? So maybe at first you'll say, I'm not going to even start. Like, the child function at such a low level, I can distinguish. Well, think about your 3-year-old nephew and then marvel about the social communicative abilities of a 3-year-old. And keep that in mind. And then ask yourself the question, am I seeing, in this child, social communicative abilities that are somewhat in the range that I'm expecting in a 3-year-old? And if the answer is no, well, then you should be considering autism as a possible diagnosis.
Thank you. There was another question, I think you talked about it earlier, about should genetic counseling, should that be a skill set that when we look at teaching and education, the psychiatrist, child and adult should have in their training?
Yes, yes. And the reason is, well, I can talk a long way. I'm not going to talk a lot, but the reason is first of like it's, and I'm not saying this because I have no stake in it in a way, but just know that if you want it or not, genetics will increasingly kind of interact or with our field, and so. You'll be getting more and more questions that relate to genetic recurrence and genetic risk and all these things. So we need to train ourselves and our future colleagues about this. And there are a couple of very good papers. I'm happy to post a link, basically saying what is the minimum amount of genetic knowledge that child and adult psychiatrists should be aware of.
Yeah, absolutely. There was another follow-up, and some of it you answered, but I think they want some clarification about which clinical presentations would you recommend that one would obtain genetic consultation? Yeah.
So if you follow the guidelines, right, the field is dynamic and in development. So the first thing to say is that guidelines internationally are not necessarily always consistent. So there are differences between certain European guidelines, between the Canadian and the American guidelines. But for autism, the convergence is quite clear. Like, all the guidelines basically say, if a child has autism, then that child actually, or adult for that matter, qualifies for genetic testing. The question is whether we as medical professionals and as doctors, would always need to ask for a genetic consultation. You can also consider the possibility to order genetic testing. But obviously you should do so if you feel sufficiently comfortable about, also counseling and interpreting the results. And that brings us back to the previous question. So we need to train ourselves. But the problem is there are not enough genetic counselors in this world to help us out with this.
Yeah, for sure. The other question that they asked was, and you touched on it a little bit earlier, about looking at schizophrenia and ASD comorbidity. How can this research, help us in that area?
Yeah, totally. So I actually removed a couple of slides, but we looked at this question of autism and schizophrenia comorbidity, and in 22q, because both autism and schizophrenia are elevated in patients with this particular genetic variant. And so the question we had was, could it be that autism in these individuals may be somewhat, let's say, precursor of schizophrenia later in life? And we actually kind of expected to see that, but we did two studies, and Bassett again was involved as well, and they were two independent studies, independent samples of patients with 22q, and both studies showed that that was not the case, that having autism as a child in someone with 22q does not increase your risk to have schizophrenia later on. So kind of showing that it's really truly pleiotropic, independently pleiotropic.
Wonderful. One last question before we end. I think you touched on it a little bit, but there was a specific question on the role of genetic testing in adults with ASD. Any thoughts on that?
Yeah, well, I hope I conveyed the message that these developments in genetic testing are pretty, recent. So there's a whole population of people that just missed the boat and were never offered the opportunity of genetic testing. So from that perspective, I would say if you have a chance to offer that to an adult, I would certainly do that. But you have to ask the question whether you think at this point a positive finding is still going to be helpful. Right, so I think in many cases you can make the argument for, but in some cases you may say, it's really not going to offer, contribute a lot to the care and the well-being of this patient. So you have to make that decision. Yeah.
Dr. Bortsman, thank you for this very engaging talk. And as I said, it covered many areas. I think it really touched on our audience and really made us more thought-provoking. And I think it'll really make us think a little bit about our own clinical practices as well, and to be more proactive with respect to genetic counseling and looking at the groundbreaking research that's going on. And thank you for your time. We appreciate it. I just want to remind our audience that our next citywide Grand Rounds is during our Future of Psychotherapy conference. The Mary Seaman lecture will be Dr. Sonia Dimajanan, who's going to be talking about expanding access and scaling up psychiatric and mental health care. And we hope to see everyone at the Future of Psychotherapy conference. We've got some wonderful speakers, Dr. Christopher Fairburn from Oxford, as well as Dr. Atul Rahman from Liverpool. So we look forward to seeing you on the 29th. Thank you, everyone. Take care. Bye.
Thank you, everyone. Thanks so much.
This webinar will end in 5 seconds.